Hi, I am Maevatiana Ratsimbazafindranahaka and I'm a PhD student from the Paris-Saclay University and the University of Andrianarimisa. Today we talk about a part of my PhD project, the characterization of the Saclay behavior in humpback whale. We all know that mammals feed their young with maternal milk. The associated behavior is referred as nursing, and from the perspective of the young, it is rather referred as suckling. These behaviors are an integral part of the mammal's reproductive system, as they ensure the early development and survival of neonates. Nursing strategies used by mammals vary from one species to another and are generally related to the developmental characteristics of the young and to the environmental constraints. Let's take, for example, the case of pinnipeds, a well-studied group in terms of nursing. For seals, the nursing is performed on land, and the mother fasts and nurses intensively during a short period of time, resulting in an incredibly fast growth of the pup. Then, the weaning occurs abruptly when the mother leaves definitively the pup to feed at sea. For sea lions, the nursing is also performed on land, but the mother alternates between nursing its pup and going to feeding trips at sea. For walrus, nursing is rather performed at sea. The pup follows continually its mother and the weaning is gradual. The humpback whale, the species we are interested in, is a marine mammal that has the particularity to undertake extreme seasonal migrations. In winter, it moves to tropical waters to reproduce and give birth, and during this period, it stops feeding. In the summer, it joins the polar zones called feeding grounds. For newborns, nursing is really important because their ability to start the long migration to the feeding grounds depends on it. It is important to know that maternal milk is the only source of energy for the calf during the first 6 months of its life. It is what will ensure its rapid growth and will allow it to undertake the journey alongside its mother. Despite its importance, the nursing behavior of humpback whales has been little studied. Most studies have been limited to surface and subsurface observations and have been based on the association between calf's positioning and nursing behavior. The description we have are therefore potentially biased and above all non-representative since a large part of the whale's activities take place at depth. The objective of my study is therefore to characterize in detail the nursing behavior of humpback whales from the calves perspective, that is, the suckling behavior, in the breeding area using CAT-SCAM tags. We conducted our study in the Sainte Marie Channel, Madagascar, one of the most important breeding areas in the western part of the Indian Ocean. We deployed CAT-SCAM tags on 3 calves accompanied by their mother. Attached on calves, the CAT-SCAM tags record videos from calves' perspective, and we used the recorded videos to accurately identify suckling events. The CAT-SCAM tags also record the acceleration in 3D and the pressure, and we use this data to characterize the behavior of the calves, that is, to determine the depth, the speed, and the body posture represented by the roll and the pitch. The pitch indicates whether the whale is pointing upward or downward, and on the other hand, the roll indicates whether the whale is leaning to the right or to the left. So we obtained a total of 11 hours of videos that allowed us to identify 44 suckling events, and we found that the suckling events were brief and rare, occurring in a sequences of 2 to 6 successive events. The average duration was 19 seconds and the suckling rate was approximately 3 events per hour. In all, the events constituted only 1.7% of the total observation time. The duration we found is comparable with suckling duration reported in feeding areas, however, the frequency is higher. The suckling events occurred mostly during dives at an average depth of 19 meters. On the dive profile here, you can see, for example, 5 suckling events, 1 isolated, and 4 successive events, and all of them are at depth greater than 20 meters. The whales might prefer to be at depth for buoyancy regulation or for thermoregulation. During nursing, according to the video recordings, the calf is generally positioned at about 45 degrees to its mother. However, the recorded pitch is much smaller. This suggests that the mother is tilted downward when nursing and not horizontal, as it has been suggested in other studies. In the videos, we also found that the calf systematically rolls to the side when it is suckling. What is interesting is that the calf actually alternates between the two sides between successive events, and this alternation is well recorded in the roll data. As you can see on the example here it is leaning to the left, negative roll, and then to the right, positive roll, and then left, then right, and so on. Statistical analysis suggests that suckling events are really characterized by a roll deviating far from zero. Compared with nearby non-suckling, the absolute roll during suckling was significantly higher, up to more than 40 degrees, whether at the surface, during descent, or at the bottom of dives. We also found that nursing is characterized by a relatively lower speed, regardless of the dive phase. In fact, this cycling behavior is so stereotyped that we were able to detect them automatically using just the data derived from the accelerometer and the pressure sensor. Here you can see an example of automatic identification with height. On the dive profile here, you have blue circles, which are the correct identification of non-cycling blocks, the red circles, which are the correct identification of cycling blocks, and the crosses, which are the incorrect identifications. As you can see, we managed to detect the majority of nursing blocks, and the false positive rate was very low. Before I finish, I would like to come back to the method we used, that is, the use of tags equipped with camera, the CAT scan tags. As I said earlier, in order to study this suckling behavior, scientists used to rely on surface observations, and they assume that when the calf is under the mover, it is nursing. In our videos, we were able to verify that the calf does not necessarily suckle when it is below its mover. Out of 59 dives during which we were able to observe the calf getting under its mover, only 25% were suckling dives. This is to say that to date, the use of the equipped with camera is really the most reliable way to study nursing with a minimum of bias. To summarize, thanks to the CAT scan tags, we were able to describe in detail the suckling behavior of humpback whale in breeding area. We found that suckling events are brief but more frequent than what has been observed in the feeding area. It is a very stereotyped behavior characterized by a high absolute roll and a low speed. We were able to develop an automatic identification method based on 3D acceleration and pressure data. It is an opportunity for the study of mover-calf pairs because it means that we can free ourselves from the visual dependence and exploit data from comparable tags without camera, for example, acoustic tags, or do nocturnal studies. So that is all I have for today. Thank you very much for your time, and do not hesitate to contact me if you have any question. Thank you.